So thank you, everybody, for joining. Good morning. So welcome to the Meet the Scientist webinar for today. As a reminder, the Meet the Scientist is an opportunity to learn more about the exciting work that the teams or investigators are doing to end Alzheimer's disease and other dimensions. And also an opportunity for you to ask questions. So feel free to start writing down those questions as they talk. I encourage you to use the chat and the QA as an options to ask the questions, or raise your hand if you want to ask the question directly. I wanted to thank again Dr. Manley, Jennifer Manley, and Dr. Adam Brickman's team for joining us today. And I'm gonna just give you a reminder of the upcoming Meet the Scientists webinars that we have. So for March, we have Meet the UCSF ADRC team. In April, we have Meet the Rush Alzheimer's Disease Center team. And in May, we have Meet the University of Kansas Alzheimer's Disease Center. For more information and all the links for registration, you can go to the website that is listed here, and you can register now in advance to all the ones until May. So once again, thank you, Ari, for joining. And now it's my pleasure to actually host and invite the teams at the University, Columbia University, to present their research. So Dr. Jennifer Manley and Dr. Adam Brickman, they are professors of neuropsychology in neurology at the Columbia University, and I'm very excited to have you here. So Dr. Manley.
Okay, great. Can you guys see this initial slide? All right. Well, we're really excited to be here today and to share some of our research with you. We've got I think all of the postdocs in the lab are going to present a little bit of their research, as well as me and Adam. And the first thing to say is that not everyone, you can't see everyone in the lab right now. Here's some pictures and names of people who are a part of the ManBrick labs. And not everyone, in fact, who is part of the group is even represented here. I was nagging them for their selfies up until this morning. So we got some at the last minute, but not everyone is represented. But these are some of the wonderful people that we work with to do our work. And thank you very much to all of you. Here's me. And I was, just to tell you a little bit more about me, I was born in Ohio. My dad was a pilot in the Air Force. He is a pilot, but he was a test pilot in the Air Force. That's this little plane here. And then we were stationed mainly on Edwards Air Force Base, which is in the Mojave Desert. And they have these signature Joshua trees, which have quite the silhouette. My mom was a math teacher. She wrote a book on teaching adults math. And when I was a kid, I was a swimmer. I was the most valuable player on my volleyball team, and I played the clarinet. During the inauguration, when the Star Spangled Banner was played, I knew every single note from every single instrument, which is a bizarre feeling because that was in high school. I have two kids. One is in college, and I have twins who are in first grade. I went to high school at an all-girls Catholic high school where my social justice bones began to grow. And I remember writing a bunch of letters for Amnesty International. I went to UC Berkeley for college, where as a summer job, I got a job at a record store on Telegraph Avenue, Amoeba Music. And I ordered the hip hop and rap records for that store throughout my college career. I went to UC San Diego and San Diego State University for grad school, and then came to New York as a postdoc. And my research is on life course social determinants of cognitive aging and dementia. across different ethnic and racial groups. I use a bunch of cohorts, including YCAP, the YCAP offspring study, the REGARD study, which is originally a study of stroke, but has incorporated cognition, and Project Talent in high school and beyond, which are high school cohorts that have now been recontacted as older adults. Really quickly, here's some data from the offspring study, which is showing different relationships between the memory of parents who are in the YCAP study and cognitive function of their adult children. And you can see that the effect of parental memory has different effects on offspring cognition depending on your racial and ethnic background. Overall, there is an effect, meaning the worse the parent's memory, the worse the offspring's memory. But this effect seems to be strongest in the non-Latinx white and the Latinx English speakers in the cohort. Okay, I'm going to pass it on to the next person, who is Justina.
Hey, hi, my name is Justina Avila-Rieger, a postdoc research scientist. I'm Puerto Rican, but I was born and raised in Long Beach, California. Of my many exciting hobbies and interests, I have always been particularly obsessed with American politics, which recently has been both an exciting and disappointing interest to have. I completed my undergrad at California State University, Northridge, where I originally majored in political science, but then switched to psychology after really falling in love with dementia and cultural neuropsych research. I then completed my PhD in clinical psychology, specializing in neuropsychology, quantitative methodology, and health policy at the University of New Mexico, and then completed my internship in clinical neuropsychology at the Baltimore VA. Now I'm in the middle of my first year of postdoc in this amazing lab, and my research focuses on using an intersectional approach to understanding sex, gender, and racial ethnic inequalities in cognitive aging. And I'll show you what I mean on the next slide. So this is data from the YCAP study. So it's well documented that women have an advantage over men on measures of episodic memory, and you can see on the first figure that we show that. But most of the research in this area has been based on primarily non-Latinx white samples, and as a result, very little attention has been paid to the fact that women of different racial and ethnic groups have different life experiences that ultimately shape different cognitive trajectories, as do men of different racial and ethnic groups, which we're showing right there in the middle plot. But by taking this variability into account, when looking at sex gender inequalities, we actually end up getting a more nuanced understanding of these inequalities. For example, we found that only white women had this advantage over all men, and the advantage over Black and Latinx women either wasn't there or was relative to men of their same race. So my overall goal with this research is to really emphasize the life course experiences of women of color and how those experiences shape sex gender inequalities in late life cognition.
Okay, thanks, Justina. Miguel, while you're getting ready, I just want to say there is one postdoc, Patrick Lau, who won't be able to present today because he's in Texas and the power went out, which sucks. So Patrick, we're all thinking about you and hoping that power comes back soon. And I bet Karen Dorsman is in that same situation. I'm trying to forward the slides. Okay, got it. There you go, Miguel.
Hi, everybody. Very excited to be here. So my name is Miguel Arce-Renteria. I'm a neuropsychologist. I'm currently an associate research scientist here at Columbia University Medical Center. I guess just a little bit about myself. So I'm originally from Tijuana, Mexico, and San Diego too. Like I think those two places are home. That's what we have up there in Tijuana. We have donkeys are painted as zebras. That's like a thing. And so besides other interests for me, I'm very, very into, I play guitar. I love guitar, especially heavy metal music. And I love cats. I have two wonderful cats. So I thought that image captures both of my loves. And I'm very, very into comic books as well. Very, very into comic books. I could potentially have a PhD in it, more so than actually in neuropsychology. So I'm very knowledgeable of it. In terms of my education, so as I said, I'm originally from Tijuana, Mexico. All my studies up until high school were in Tijuana. Then I did college in San Diego State, then came out to New York for my grad school in clinical psychology at Fordham. And then after completing my PhD, I joined the lab in 2017 at the Columbia, at the Taub Institute. Previously, during my grad school work, I used to do neuro-AIDS, HIV/AIDS disparities research. And then what's great about joining the lab is now I'm focused on disparities, but in cognitive aging. And since then, like I mentioned, I joined as a postdoc in 2017, now lucky enough to transition to an associate research scientist position through a K99 R00, which my main focus of my funded work is looking at bilingualism as a factor of resilience to cognitive aging, specifically within Latinx populations. And my approach is really into breaking down different aspects of bilingualism, such as, looking at age of acquisition, language use, and other factors. But similar to what Jan had mentioned, I'm also very lucky to work with other databases looking at disparities in those studies, such as primarily obviously with Offspring. All my work comes from Offspring, but then the Regard study as well, and with the Mexican Health and Aging study as well, like in terms of how to develop diagnoses for MCI. And for the next slide, I'll summarize a little bit of my results, some of the recent stuff that you may have seen at INS this year. And so here, this is with the Offspring study from a sample 185 English and Spanish bilinguals. And so a big thing is I focus within bilinguals, excluding monolinguals, rather than doing a bilingual monolingual comparison. And what you're seeing here is we were evaluating the effect of parental history of AD on cognition and bilingualism in terms of frequency of language use, which we divide the groups bilinguals into active bilinguals, meaning they use their daily language they use both languages every day, versus passive bilinguals, meaning they have knowledge of a second language, but they don't use it as daily as active bilinguals. And as you'll see here, you'll notice that the active bilinguals were performing better on a task of processing speed, but were really not so strongly associated with set shifting, working memory, or attention. And then similarly, in this sample, there was no strong effect of parental AD on cognition, which was surprising because in the whole sample of Offspring, we did find that negative association. And then the next 2 clicks, I think. Two more clicks, then you'll see us evaluating whether the effect of parental AD, if that differs by bilingual active passive status. And you'll see here on the top, on your right, on the top, we have the effect of parental AD within passive bilinguals, and then parental AD in active bilinguals below, across these 4 different domains of executive functioning. And you'll see there's no reliable differences between the effect of parental AD in the group. And all of these models adjusted for age, sex, gender, years of education, immigration status, and parental education. And these are still preliminary results because we were working with the data that was available for our language history. But currently we are still collecting more data. But I think that provides a broad overview of the kind of work that I'm doing.
Hello, everyone. I am Indira Turney, and I'm a 2.5-year postdoc in the ManBrick lab. So Jen, if you can click on the first video. I'm very active on social media, so this is a video I made for mentees. Well, we can start with the crocheting over here. I do a lot of crocheting. I recently got into that. These are gifts I made for a friend over the Christmas break for her cats. And then the second video at the bottom. And I made my friend
this is my sign. This is where I live, and what I like. This where I'm from, and what I do. This what I love. How about you? I know you want.
So that was a video that I made for mentees online just to like introduce myself. So as it shows, I love my dog Buddy. I'm also from the island of Dominika, which is the first map after this video at the top. And then I moved to the Virgin Islands, specifically St. Croix, which is the second map over here. I have a guitar in here because I've been inspired by all the musicians in the lab, but I've played it probably 10 times and I've bought it since last May. But Miguel has promised to teach me, so we'll see, and Justina. And so when I moved to the Virgin Islands, this is where I finished high school. And then I attended the University of the Virgin Islands, which is the only HBCU outside of the mainland US. I majored in psychology there, which then I moved during the summers while I was an undergrad. I did summer research programs, so summer research opportunities at the University of Iowa, as well as Ohio State University, which then led me to do a pre-doctoral program at the University of Pittsburgh for a year after graduating from the Virgin Islands. And then I did my PhD in cognitive neuroscience at Penn State, which then led me now to Columbia University Medical Center, where I am here with Jen and Adam, and my research focus using both YCAP and offspring data mainly to look at the social cultural factors that affect brain health, specifically across different ADRDs, and specifically also looking to transition more into also looking at biological aging and how all these things merge together to explain health disparities across the spectrum. And so
This is my age, this is my time, this is what I live, yeah. What I like, this one from.
I don't know why it's not moving, okay. And so the next slide here, just looking at one of the projects that showed looking at health disparities across the offspring cohort, so the younger cohort, which are the children of the YCAP participants. So in here we have looking at white matter hyperintensities and cortical thickness across cohorts. So in blue we have the YCAP participants who are older, and the red we have offspring participants who are younger. For white matter hyperintensities, we see that in general, older participants, so in blue, had higher white matter hyperintensities compared to offspring cohorts. We also noticed race disparities where Black and Hispanic participants, both in older cohort and the younger cohort, had a higher white matter hyperintensity volume compared to the white participants. And then for cortical thickness, we see what was expected, that the offspring cohort, so the younger cohort, had higher cortical thickness compared to the older cohort, as well as showing disparities in the older cohort. So in blue, we see that Blacks and Hispanics have less cortical thickness compared to whites, but this pattern was not observed in the younger cohort, where we see kind of more of a similarities in cortical thickness across the younger participants. So all in all, we're showing here that white matter hyperintensities, which no one is born with, is obviously even apparent even in the younger cohort, showing that there are other factors explaining some of these racial differences. And so future work will look into what those social cultural factors are. And so that's just a glimpse of some of my work, and happy to talk more about it.
Hi, I guess I'm next. I'm Lok Kin Yeung. So I'm a postdoc with I work mostly with Dr. Brickman, but also a little bit with Dr. Manley. So I grew up in Toronto, as this picture of the skyline shows. My family migrated from Hong Kong when I was a kid. They give you a little citizenship card when you become a citizen. So I'm stuck with that for life. That's what the Canadian government will think of me for the rest of my life. I've been spending quarantine with my cats. You can see them there. I spend a lot of time on historical grand strategy games. And I guess like my research interests were really sparked by this question. How does the brain think? How do we remember things? Like I remember taking a class in college where like the professor said, okay, well, this is exactly how a computer represents a picture, a text file, a video, and so forth. And we have absolutely no idea how the brain does that. So to kind of pursue this line of work, I started working with Dr. Brickman as an undergrad, then pursued my graduate work back home in the University of Toronto. And I've been back in New York for the last few years, again, working with Dr. Brickman as a postdoc. So I feel like my work is kind of bifurcated into two pieces. One piece is working on this large study of cognitive aging using online virtual testing called Cosmos Web, which we're about wrapping up right now. Very excited to kind of delve into the data. And the other line of my work looks into how brain changes in the hippocampus and neighboring regions of the brain affect different cognitive abilities. And so I'm going to show you a little bit of that data in the next slide. So I've kind of cut and pasted two pictures from some of my papers here. So on the left, it shows like a manual segmentation of this part of the brain called the entorhinal cortex that I've been studying. And you can kind of see the cross sections kind of going forward in the brain. And so in some of my work, I've shown that this relates to a wide number of different cognitive measures that you can see at the top right. I've also shown that cerebral spinal fluid measures of amyloid, which is a biomarker for Alzheimer's disease, is also related to memory, not surprisingly, but also surprisingly, even in people who are quote unquote clinically negative. So this is kind of a surprising thing because it suggests that even very low levels of amyloid might start to change your memory even before when we think you begin to change.
Hi, my name is Vonk, and I am a collector of degrees. And I'm a neurolinguist and an epidemiologist. I am an associate research scientist at Columbia University. I have been a ManBrick since 2017. And I'm also affiliated with the UMC Utrecht in the Netherlands. And that's a silhouette of my country in case you've never seen the Netherlands before. And I play basketball, I run, and I snowboard. And I think it's very hard for me to get through a day without at least a gallon of tea, hot tea, and 3 different pieces of fruit, which, you know, was a little bit problematic in the beginning because Adam hates bananas, but we got over that and the mentor-mentee relationship survived. I got 2 bachelors and a master's in neurolinguistics from the University of Groningen in the Netherlands. I got a master's and a PhD at the CUNY Graduate Center in New York in speech, language, hearing sciences. And I got a master's and I'm in the process of getting a second PhD in epidemiology at the University of Utrecht. And my main interest is language in the brain. I was obsessed with that since the early beginnings of my academic career. During my early career, which I'm in the middle of, but I spent some time as a visiting researcher at Newcastle University in England and at the UCSF Memory and Aging Center in San Francisco. And in the last 2 years, I fell in love with epidemiology. So currently my research is at the intersection of epidemiology, neuropsychology, and neurolinguistics. And my research is now funded by a K99 R00 award and a VENI award from the Netherlands. And my research is focused on word meanings and how they're lost in the earliest stages of Alzheimer's disease. So what I do in a little bit more detail is in the next slide. So what I do is I leverage existing neuropsychological test data, and by using my linguistic background and my linguistic knowledge, I develop alternative and more sensitive metrics to detect the earliest signs of cognitive decline in the preclinical phase of Alzheimer's disease. And you can see some promising results in the graphs on the bottom right, that these novel metrics may indeed capture valuable information that traditional measures don't. So in this graph, you see that using these novel metrics can distinguish APOE4 carriers from APOE4 non-carriers, so a higher genetic risk for Alzheimer's disease, while the traditional metric in the same sample does not cannot capture this difference between APOE4 carriers and non-carriers. And an important goal of my research is to deconstruct cultural and demographic effects on these novel metrics, like education, sex, gender, and race, ethnicity, because the aim is to develop these metrics into a clinical tool that we can use to investigate and identify individuals at high risk for Alzheimer's disease, and to do this for individuals from all backgrounds. And I'm happy to answer more questions about my research.
Hey everyone, I'm Jeff. I'm a mechanical engineer. I love building things. I'm originally from Arizona, and as you can see by the photo, for all of those of you in winter climates, the desert does get snow, but it only lasts for about maybe a couple hours. But this is one example of it. But I love photography. I love music, and I think a surprising fact about me is that I got into skateboarding recently, which is always a bit of a dangerous endeavor, especially in your 30s, but it surprised everyone, including myself. some of my early work was in aerospace and in cells, and that kind of brought me on an interesting pathway to then get to eyes and studying human eyes, which then brought me to the brain to look at computational models of cerebral blood flow and vascular aging. This was at originally University of Arizona, then at Berkeley. And then now I looked at the vascular effects of aging and looking at kind of the vascular contribution to Alzheimer's, looking at blood flow models and trying to capture measurements within individuals for how blood flow changes. So this got us to the next slide here, looking at a recent example of vascular changes in the form of COVID. So some of our studies here look at different pathways to damage and some of the neurological effects of COVID. So that can include everything from central nervous system changes to peripheral nervous system changes. And then we're also interested in how does this affect cognition? And then, so here we look at clinical COVID-19 survivors and see what kind of changes happen over time. Yeah, thank you.
Hi, everyone. So my name is Dominika Seblova, and I'm a social epidemiologist. I am originally from the Czech Republic, in case you also did not know how Czech Republic looks. That's the map on the left. And I have a lot of interests outside of work. I love to mainly go hiking and disappear offline for at least a week at a time. I like biking, I used to play volleyball, I like baking, and a lot of, sometimes I draw. And I guess my other hobby is actually country moving and continent hopping, because as my education shows, I have been sort of back and forth between a lot of places. So I left Czech Republic when I was 17 to attend my last 2 years of high school in an international setting in Italy through United World College system. And then I went to United States. That's my first cross-continent hop to do my bachelor's in anthropology. And then I decided to go back to Europe, to Sweden, and went to Stockholm University for my master's, focusing on health inequalities, and my PhD at Karolinska Institute. So during my early career that I still believe I'm in, I have been focusing on inequalities in healthcare, in health in general, and more specifically looking at how education affects health and cognition specifically. And now I'm at Columbia University, and I mainly use project talent data that Dr. Manley has already mentioned. And I'll show you some of my ongoing projects in the next slide. So my main work in the project talent data has been looking at high school quality. So in project talent, there have been students in high school who completed individual questionnaires, but also the principals have answered questions about the schools. So we have been trying to use those answers from the principles to derive a measure of high school quality. And in the top left graph, you can see that we specifically look at racial ethnic differences in the quality of schools people attended. And then I showed 3 different graphs, and I will not go through them, but I just wanted to mention the projects I have going on. And that is, we are looking at later life cognition, and that is the top right graph. And so we so far found that in 70s, so 58 years later after high school, attending higher quality school was overall positively related to later life cognition, but the returns were lower for non-Hispanic Blacks in language abilities. The bottom left graph is actually looking at self-reported cognitive impairment, and again, there the returns are to the blue dots, which is non-Hispanic whites, but other racial ethnic groups, especially non-Hispanic Blacks, did not report lower odds of self-reported cognitive impairment when attending higher quality high schools. And the last project is actually looking at mortality. And there we find that only those who identified as other racial ethnic groups had lower mortality when attending higher quality high school. So this is my research right now, and we're trying to take it further and see if different aspects of high school quality matter for later life cognition and health. And yeah, that's it for me.
I guess this is me. Hi, everyone. I'm Adam. I am the brick in the ManBrick labs. This is a little bit messy here, this slide that I made this morning. I'm just displaying the different countries that I've lived in up on the left, mostly during my early childhood. But then as you can see by the map of the United States colored in purple, I've lived all over the United States, including New York and Ohio, but quickly came back to New York. I love early mornings. I love art. I love coffee. Earlier in my life, I thought I was going to be a professional musician until I got to college and realized I wasn't good enough. But I've always loved indie rock and punk, primarily from the 90s. I really like interspecies relationships in animals, like this elephant and ostrich. It's one of my favorite things when different types of animals get together. I like traveling. I like the water, but I don't like being in the water. I like being near the water. Just to highlight some of my personal life, I started off collaborating with Molly Zimmerman. We were training together at Brown, and then we collaborated more romantically and had this child and got married. We also had these 2 dogs. We have a ton of animals, but those are 2 of them. I've trained, I went to college at Oberlin. I was a research assistant at Mount Sinai, which really was an inflection point for my interest in neuroscience and aging and dementia. I continued working there for my dissertation work while I was a PhD student at CUNY. I did my clinical training at Brown, and I've been at Columbia since postdoc. This is our lab seal. I can explain this some other time. My favorite things professionally really are collaborating. Here's my #1 collaborator, Jen, mentoring and being mentored. And oftentimes the collaborators, mentors, and people I'm mentoring are playing all those other roles for me too. So it's really awesome. I do most of my work incorporating brain imaging in ongoing or large social or large community-based studies. So we try to look at the intersection of vascular and structural and AD biomarker changes and community health and social determinants of health in community-based samples. So going on to the next slide. If possible, it's not advancing. So I thought it was important. No one wanted to show this, but people wonder what the lab organizational structure, because we're sort of 2 labs coming together and forming a meta lab. So this basically is the diagram we came up with to highlight how the ManBricks really come together. I think it's pretty clear. Next slide, Jen. And we do a lot of different work, but I wanted to highlight this recent paper that I'm really proud of. I'm particularly proud because Crystal led it. She did this beautiful job. We were interested in understanding whether supravascular changes promote tau pathology in Alzheimer's disease, and it's sort of a translational neuroscience paper where we first demonstrated that there's a reliable relationship between markers of cerebrovascular disease and tau pathology, particularly in people who are symptomatic. But of course, correlational studies in humans get us so far. So we designed this animal experiment where we did sort of an animal model of white matter hyperintensities by doing transient hypoperfusion by occluding the MCA in mice. And what we showed, you can see in the top right panel, is when we occlude the MCA transiently, we can induce this white matter damage. You see this myelin basic protein markers in the down in the occluded mice. But fascinatingly, that induces hyperphosphorylated tau pathology throughout the ipsilateral cortex, and particularly the hippocampus. And then we were able to show in the mice, we did little lumbar punctures and venipunctures and assayed their CSF and their plasma, and showed also that the same biomarkers that we see in humans, the same assays, can detect the tau pathology in the occluded mice. And so what I think we at least proved to ourselves is that you can induce Alzheimer's pathology, tau pathology in particular, through a vascular pathway, and it's entirely independent of amyloid, and it's causal. So we're very excited to follow up this work. This is definitely a direction we're going to go in some more, and kudos to Crystal, who really did a beautiful job on this work. That's all I got.
All right, and that ends our presentation. We are very ready for questions. I see people had some comments. I know that Jakil asked me to say a little bit more about the YCAP study. I can do that now, or if there are other questions. Oh, I see there's a Q&A. Oh, Annie, okay, all right. Well, why don't I just quickly, I'm seeing the questions. Why don't I just quickly talk about the YCAP study, if that's okay with everybody, and then we can answer the questions that are in the chat. So the YCAP study was established in 1992. Richard Mayu and his colleagues used Medicare eligible lists of Medicare eligible people to find the names and addresses of folks who lived in the census tracts around where the Columbia University Medical Center is, which is in Washington Heights. And it's a very, it's an incredible neighborhood. A lot of people from the Dominican Republic have immigrated to live there. There's also Puerto Rican and Cuban people. It's a Spanish-speaking neighborhood, so really everything, you know, if you don't speak Spanish, you have a little bit of difficulty making your way around in Washington Heights. But there's also buildings, naturally occurring retirement communities that contain a lot of non-Latinx Black people, many of whom have lived there for a long time, and also non-Latinx white people coming from a whole bunch of places, including Hungary and Poland and Germany, and they came to the United States to escape the Nazis. So there's a lot of diversity. Their cohorts were recruited in 92, 99, and 2009. So over 6,500 older adults have participated. And what Adam and I did in 5 years ago was got a grant to study their adult children. So we're recruiting people age 30 and over who are the children of the YCAP participants. And we're looking at this cohort as sort of revealing this intergenerational cohort as being really promising to revealing some of the life course mechanisms of disparities in dementia and cognition. Is that pretty good, Adam? Okay. Annie writes, any tips for trainees who are interested in conducting health disparities research, like training, mentoring, workshops? That comes from Annie Reyes. Anybody want to take that?
I would say, I guess, just in terms of the things off the top of my head, I would suggest, I think, a few different things. I think definitely reaching out to potential people who do, you know, disparities work, so you can start collaborating to some degree and then get exposed at it from that level, like either at in-home or reach out to other institutions as well, like ManBrick labs, perhaps, I don't know. But besides that, I do know there are training opportunities. I can't remember off the top of my head, but I know, like, for example, for more advanced, I think at least for postdoc and above, the NIMHD offers a disparities like workshop, and I think APA does that as well, which I think is available for people who are not who are still in grad school. But so there might be things like that as well. But off the top of my head, that's what I think.
Great. Yeah, I think you know, here's my take on it. You know, my take on it, Annie, soon to be Dr. Reyes, is that you can't really do health disparities research if you have a small clinic-based cohort. So that's really what you want to look for. You know, one of the stories that I tell is that when I was in San Diego, I was working in HIV Neurobehavioral Research Center. And, you know, as HIV research started to, you know, get more diverse participants, it was hard. It was very hard in those days to recruit people. There was just a big lack of trust. And so, lack of trustworthiness, more importantly, among researchers. So what happened was that when I was looking for a place to do my postdoc after internship, I just went to, you know, Ovid, what it was then, PubMed, and looked up neuropsychology or neuropsychological and African American. And I looked for the study that had the most people. And that was the YCAP study. And I happened to know one of the neuropsychologists, Diane Jacobs, who worked at Columbia at the time. And you know, they weren't doing, other than the genetic work, looking at the differences in the impact of ApoE, there wasn't a ton of questions being asked at the time about the ethnic and racial disparities in dementia. So, you know, I ended up being able to do a lot of this work. But my point here is that small select cohorts that are, you know, select themselves to become, you know, to go into clinics really aren't great for examining the reasons, pathways, or mechanisms of health disparities. Alex Wiegand asked a whole bunch of questions, but they've gotten answered in the Q&A. Adam, did you see those? They're under the answered group. I'm just going to ask Udell Holmes.
I did.
Sorry.
I typed out answers, but we can talk about that.
Oh, that's all right. Yeah, maybe you can pick one to talk about real quick.
So Alex asked a question of Miguel. Miguel, do you want to take it? I don't want to hog.
Yeah, sure. I could quickly respond. You know, right. Alex asked about why do we think we didn't see the association between active bilingualism with better executive functioning, specifically with set shifting. You know, and that's a very good question, because I think within the literature of the bilingual advantage, specifically with executive functioning, that's always been within bilingual compared to a monolingual group. You know, from what I can remember, I think there's been either one study only looking at active bilingualism, and it looked at a different kind of executive functioning task, more of a response inhibition type test, not necessarily a set shifting task. And so, you know, why are we not finding that impact? I think there could be a different things from the differences between the tests being used. It could just be that active bilingualism does not lend itself to such a widespread benefits in cognition, such as in executive functioning. It might be like we were mentioning, obviously, as well, a preliminary sample. We'll see with the sample growing. Another important thing, as it was, I didn't mention early, is that this is the offspring study. So the sample is primarily middle age. And so it could be that the benefit to cognition may be noticeable much more later in life. But another thing, and so from what I mentioned at the beginning in terms of my approach, is there might also just be other linguistic characteristics that might be important to keep in mind. You know, they're active bilinguals now, but it might be important to know how proficient are they, for how long did they acquire the language, and maybe a combination of these factors can lead to benefits in executive functioning, specifically set shifting.
Thanks.
I quickly just ask a follow-up questions.
Yeah.
Miguel, I was wondering also, is you happen to study anything with verbal fluency, or did you notice any change with verbal fluency in particular?
We have not looked at that yet with this data set yet with verbal fluency, but it's definitely something we're interested in looking at, especially given as like with the literature, bilinguals tend to perform worse on verbal fluency measures compared to monolinguals. It'll be interesting to see how that works in terms of these within aspects of bilingualism.
Okay, Udell has two questions. Gosh, Udell, why don't we have Jeff answer the second one? Can you see it, Jeff, under the Q&A, or do you want me to read it? Okay, you can see it?
Yeah, I'm just reading it now.
Okay. Yeah, you could just read it because maybe there's people who can't see it who are on the call.
Yeah, so it's asking about the potential neurological issues that can come with COVID. Even though the virus is novel, is there a concern in the field of neuroscience that we might see some of these survivors being at greater risk for developing Alzheimer's or vascular dementia? I think, I mean, from what we're seeing, our hypothesis is it's likely. I think it does depend on the severity of disease, especially for people who have really strong inflammatory responses, or potentially a lot of blood clots or other kind of secondary organ damage. So it does seem like something that we're interested in looking at in the future. But as of right now, there's not a lot of long-term outcomes yet. I mean, the most we have is like maybe 6 months or so, but it definitely seems like there's a couple different pathways that are all independent that could converge to the same points. And that's, yeah, it's definitely a risk for the future.
Thanks. Last week, Jeff gave a whole talk at our cognitive neuroscience seminar about some of the potential pathways for COVID infection, including inflammatory pathways to influence the amyloid and tau and vascular cascade that can lead to Alzheimer's dementia. So that was great. Christina Roman, given the relationship between life stress and neuroinflammation, how do you think significant life stressors like systemic racism might impact cerebrovascular disease and cognition through a neuroinflammatory process? We have been thinking about this. Nika, do you want to give this one a shot?
Sure, I can give this one a shot. So actually that has been a question of interest to me, and I have put in a proposal now to look at using the offspring cohort, looking at measures of discrimination and inflammation and how does that affect cognition, with potentially extending it to cerebrovascular pathways over the time. And I think a lot of the literature up to now does show that, stress, including discrimination, do affect inflammatory processes, but a lot of it is actually measured by peripheral blood markers. And then it's hard to know how directly it relates to brain in itself. So this is what we're hoping to do in the future. There's also really intricate relationships between, for example, reporting of discrimination and stressful life events, which is also related to one's education. So those who are more highly educated tend to report more discrimination, even though that might not necessarily be always capturing. So there are some measurement issues in this field, but we're hoping to report on this in a soon future.
So I just wanted to bring up, just highlight something here, which is sometimes when you talk to people in the field or you read the literature, sometimes things like the social determinants of health and vascular contributions and medical and neurological measurements are often siloed as distinct fields. And I think one thing that's important to highlight about our lab or meta lab, however you want to think about it, is that we're really in a position where we can integrate biological measurement with social measurements and really start understanding how some of these social determinants of health interact or promote some of the biological changes we see in the face of aging and disease. So it's been really, I think it's a really powerful and unique aspect of our particular program. Jen, sorry to hijack here, but Yakil mentioned in the chat asking if there are positions open for postdocs. And I would say, anyone interested in collaborating or working with us in whatever capacity, reach out, let's have a conversation. We don't have a postdoctoral training grant per se, but we have grant support that can often support new postdocs. And also there are a lot of interesting ways of funding postdoctoral experiences. So I would say, generally speaking, yes, there are always potential training opportunities in our groups.
Can I ask a follow-up questions there? Like, it seems to me like design, everything that you present is very impressive. I found very unique too. Like, is there anything that is holding you against applying for a T32 postdoctoral grant or something like that alone? You know, I don't know,
So
an idea.
Yeah, Kayla, it's something that I'll let Jen answer too, but it's something we've talked about a lot. And our division used to have a postdoctoral T32 program. I'll speak for myself. I think that the T32s are great, and I think we have the infrastructure and the themes that could support a T32. The one concern that I have is the salaries of the T32 grants in a city like New York. You know, those NIH set postdoctoral salaries are a little bit tricky to live. And so generally speaking, we've been supporting postdoctoral fellows either on a line, on an R01, or with diversity fellowships against the different grants that we have. And that's been our approach. But, you know, I think, yeah, I think we have the infrastructure, and effectively we sort of run a T32, although it's not officially a training grant %.
Yeah, I don't know if any of the fellows want to say anything about that. One of the things that I thought I would mention, just in general, you know, given that this is a Meet the Scientists and Meet the Lab thing, is that Adam and I talk a lot about you know, trying to, you know, protect the well-being of the people who are working with us, and part of that is, you know, making sure that people have enough money to live in New York City, which traditionally has really not been all that easy. So that's part of it. The other part is just, you know, we think a lot about how to support people, you know, in the, you know, to get what they want. One key part of our mentorship is individual development plans. Everyone has a different pathway, and we really try to honor that and work together on that, make expectations really clear, and also make sure that, because Adam and I can't give you know, everything to everyone, we have a whole, you know, university and a whole world of collaborators that we make sure that our group has contact with. You know, I don't know if any of the folks want to say anything.
Yeah, I can add to that. That also connects to what Yukil just mentioned about that all of us have very different research interests, and there's a lot of different topics that we're looking into. And I just want to highlight the space that Jen and Adam give us to develop our own research interests. So you might have heard in our little pitches that, for example, Justina and me blurted out words like, I love what I do, and this is like my passion. And that's because we got the freedom to explore our own interests. And of course, we get very great and weekly mentorship from Jen and Adam, but they have actually encouraged us to use our previous training background and combine it with the new knowledge that we learn in this lab and the research that comes out on a daily basis to develop our own research lines. And I know that's not the case in every lab in academia, and I think that's really a huge strength of us, and also why we are such a healthy and happy lab, and everybody loves the research that they do and the research that other people do, and we collaborate a lot on that.
So thank you, Yet. Lisa Barnes asked a really hard question. I've been thinking about it as we're speaking, like my mind's going two ways. Like how am I going to answer this question? She asks, for people interested in life course disparities, how do you guard against your intergenerational work in the offspring study being used as evidence of genetic determinants of disparities? Does anybody want to take a shot at that? Nobody? Okay, well, here's my initial answer, and then and other people can jump on. And I know we only have a couple minutes, but Lisa, one of the things we're doing is measuring genes. We're getting GWAS on the offspring in the YCAP cohort, and we're also trying to, really honor that health disparities framework, which has different levels of analysis, not just biological or genetic, but also social, psychological, behavioral. We're measuring a lot to do with the context that people are in. And I think one of, the family design is actually really powerful because analytically you can, actually have a variable that accounts for the relatedness of people, and then look at the effect of the environment on some of the outcomes you're looking at once that latent sort of relatedness variable, or the hereditary variable, if you will, is accounted for. So we think that actually this intergenerational design is really great for looking at some of the environmental influences. It's my belief, based on what we've seen so far, and also just looking at YCAP, where we have people who have immigrated here, people who have reported their childhood socioeconomic status, where we have looked at genes, where we have accounted for ApoE4, which is the most in any group. Although the strength of its relationship to the cognitive outcomes differs across race and ethnicity and across the populations, where we've accounted for ApoE4, the social factors seem to be much more powerful in determining someone's later life outcomes than the genetic factors. So I think just to sum up, I think we're trying to measure as much as we can and look at this as a holistic sort of multi-domain process, rather than a single genetic process that's going on. Any other thoughts on that?
I was gonna say something similar, but to add to that, like, I think because we're also finding in certain, especially with the biomarkers, different relationships across offspring and YCAP participants, I think it's a perfect, sample where it kind of disproves some of the things of like, well, once this is your genetics, you're doomed kind of thing. So I think it's a really great data set to look at the different influences of environmental factors and all the other measures that, even though we don't have a strong relationship with ApoE in some cases, but maybe over time, as we look at other genetic markers, that the ones that do have an effect, we can probably see how it's really more about environment and not so much just genetics. So I'm really excited about that.
Justina, I think also Justina's work is looking at what was thought to be biological effects of sex gender and finding that those differ across group as well. There was a question about data access. What was it?
Somebody asked about if they're interested in some of the data that we work with and they're interested in working with it, how they could go about it. And I think that would be great to bring up.
Yes, they can email. The thing I would say is that they can email me, and I can get them hooked up. If I don't answer my email, email me again, or email Adam to ask me to respond to the email. That sounds good.
Or really email one of the postdocs and we can.
Yeah.
So just a quick, there is a formal data request access, but working with one of the core investigators always helps demystify that process. So that there is a mechanism to request data. I think we're out of time.
Yeah.
Yes, we are out of time. Thank you so much. It has been great. Before we say goodbye, I just want to give Miguel the opportunity to remind everybody about the NIA workshop that you're organizing. I think that many of the people that are listening will be very interested in joining.
Yes, we'll do. And I forgot to mention it on my slide because I'm also very happy. I've been involved with the PIA, the disparities, diversity and disparities PIA right now, the co-chair of the bilingualism SIG, bilingualism, literacy and languages SIG. But very excited that I was asked to help plan an NIA workshop on bilingualism and cognitive reserve. And the workshop is going to be March 2nd and 3rd. And so there's an Eventbrite link. It's on Twitter. I've shared it. Please, if you haven't seen it, if you're interested, it's obviously free to register. It's 2 days, a wonderful group of people presenting talks on this issue that, hopefully can lead to really important discussion. So you'll see an expanded, much expanded presentation of what I talked about today. So email me or on Twitter or whatever, and I can give you the link to sign up.
Thank you, Miguel. And thank you, everybody, again, Dr. Manley, Dr. Brickman. Thank you so much for joining us. And the next one, March 29th, to meet the UCSF team. So thank you, everybody. Have a good day.
Thanks for having us.